I'm going to skip through this. We should know the difference between the three types of muscle tissue at this point. It was in various chapters so far. These are the properties of muscle that I'm not just going to read out to you. Excitability is only in muscle and nerve, but contractility is only in muscle. So that's just very exclusive property that only muscle tissue has, that no other tissue type has. Where excitability, of course, action potentials can travel, or we can also say nerve impulses, can travel on the plasma membrane of muscle cells as well as nerve cells, neurons. And of course, you can stretch them and they go back to the original length. That's what these two properties have to do with here. All right, most of you know this from lab as well, so I'm just going to just briefly mention that the epimysium is the connective tissue covering over the entire muscle. The perimysium is the connective tissue covering around the fascicles, which are these little sections. If we enlarge one fascicle, we can see that a fascicle is made up of muscle cells that we call muscle fibers. So from now on, a muscle fiber is a cell, and endomysium surrounds the muscle fibers. All right, I think we're okay there. So muscle fibers, which are cells, are unique to other cells in the body. So a lot of their parts, which when we labeled a cell back in chapter, what was it, 3 maybe? They're given special names when we get to muscle cells or muscle fibers. So we don't call this the plasma membrane, we call it the sarcolemma. That's a big thing. Inside the muscle fiber, we have these cylindrical structures. Each one is called a myofibril, and they're packed in there. All these cylindrical structures, myofibrils, you can see mitochondria there as well, are inside this muscle cell or this muscle fiber. Now, if we just look at one myofibril, which is down here, this is just one myofibril now, so we're highly magnified. We can see that it's made up of myofilaments. So there it is, myofilaments. There's thick filaments and thin filaments. In purple, we see the thick filaments, and that's called myosin. In green, we see the thin filaments, that's called actin, and they overlap each other. So the thick filaments are overlapped by the thin filaments on either side. So actin is on either side of myosin, overlapping, and this Z-disc kind of separates section of myofibril. So in the middle, from this Z-disc, which is in the middle of all these actin fibers, to the middle of, or to this Z-disc, that's one contraction unit, and that's called a sarcomere, and that's the structural and functional unit of skeletal muscle. Striated is another word for skeletal muscle. So the way this sarcomere contracts is by overlapping more and overlapping less, which is called the sliding filament model. So actin and myosin will slide against each other, which will move these Z-discs closer together or far apart. So each myofibril has many sarcomeres, right? Many units of contraction. And from Z-disc to Z-disc is just one. And we could keep going from that Z-disc to one that would be down here is another one. From that one to the next one would be a third, and they're just arranged in series. Need to move this little thing. I'll move it up here. And then we have these terms, these bands and zones. Well, we already named the Z-disc. But if we look at the entire muscle fiber, we can see it's got that striated appearance. It goes from light to dark, etc. And that's because of how these myofilaments, the actin and myosin, arrange themselves. So right in through here where there's no myosin and only actin, it looks a lot lighter because there's less protein filaments there, less myofilaments there. And we call that the I band. So the I band is this light band. Where myosin is present, all in through here, and overlapping actin as well, this whole area, the entire length of the myosin, the thick filaments, that's called the A band, and those are the dark bands. So that's how we get the light band, dark band, light band, right, and so on, which are I bands, A band, I band, right, alternating, and so on. Easy way to remember is that the letter A is the only vowel in dark. The letter I is the only vowel in light. So kind of worked out nice that way. That worked out nice. The Z disk exists in the center of one I band, right? The center of a light band, and there's the center of the next light band. So there's the Z disk. The M line is just kind of holds the myosins together. That's these proteins here. The H zone is inside the A band. So inside the A band, there's an area where there's no overlap. See where the actin only goes so far? It only stops there, it stops right there. So that area where there is no overlap is the H zone. All right, so now we zoomed in on just one functional unit, which we call a sarcomere. There's our Z disk. By the way, Z line is the same as Z disk. There's the other Z disk. It's in the middle of the light band. To the middle of the next light band, there's our sarcomere. There's our overlapping actin and myosin. We see a little bit of overlap. And if we look a little closer, so let's take myosin, the thick filament, and enlarge it, we can see all of these little arms coming out of it. They're called myosin heads, and there's just one of them. Now let's enlarge or magnify the actin, the thin filaments, and we can see it's these two chains that kind of weave around each other. That's the actin, and if you look closely, we can see there's all these little holes in each little piece of protein in the actin. Those are the binding sites. That's where this myosin head wants to insert into these binding sites on actin. And when the myosin head binds to the binding site on actin, it propels actin towards the center of the sarcomere. So if we look back here, you see the tiny little myosin heads. When they grab onto actin, they're going to pull it towards the center of the sarcomere from either side towards the center. And that will bring the Z disc closer together as there's more overlap between the thin and thick filaments. Another very important point, this myosin head cannot bind to actin because the binding site is blocked by these yellow, well, they're not actually yellow, the artist made these yellow proteins. You see this one right here. I guess it's more orangey yellow. And there's another one right here. It's a little ball. These proteins are called troponin and tropomyosin. Those perform, I just added this before we started today because I saw it didn't say it there. So troponin and tropomyosin perform contact inhibition. They're blocking, right? They prevent the myosin heads from binding. They're in the way. So if that myosin head wants to go up and grab actin to slide it towards the center of the sarcomere, it can't do that because tropomyosin, this long orange-looking protein, is inhibiting it from doing so, right? Contact inhibition. And troponin, I can call them the T proteins. They both are the ones that start with the letter T, is part of that complex. So that troponin, tropomyosin complex prevents myosin from binding to actin. Of course, if we want to contract a muscle, we need to get those proteins out of the way, right? So the myosin head can bind. So that's going to be the next step. But here we go. When a muscle is relaxed, tropomyosin is positioned in such a way that the binding sites for myosin are blocked. So this is a nice illustration. It shows the Z discs, how far apart they are when the muscle is relaxed. Look at the amount of overlap between actin and myosin. Now here is a contracted sarcomere, right, where there's a lot of overlap between actin and myosin. So none of the fibers change length. Actin is always the same length. Myosin is always the same length. The difference is less overlap and more overlap of the fibers. And that's what the sliding filament theory is actin and myosin, or the myofilaments, as they're collectively known, sliding against each other. All right, I'm going to take a quick peek if there was a question. I didn't think so. Okay, good. Oop. Someone have a hand up? No? I thought I saw a hand go back down.
Yes.
Oh, hi.
Hi, Professor. I have a question for you.
Okay.
For the test, will there be any labeling of the structures?
No. All the label, we do so much labeling in lab
Okay.
that I kind of save it all for lab. It's just we're kind of labeled out, right, with
Okay.
labs. So yeah, so for the lecture unit exams, it's all text. It's all words.
Okay.
I know I
Okay.
was kind of showing you what the structures look like, and that's so you just kind of get a good visual what's going on, but there won't be any pictures.
Okay, great. Thank you.
Yeah. Uh-huh. All right. Now we're going to keep going here. So we're talking about skeletal muscle, of course, which is voluntary, meaning we consciously control when to contract our skeletal muscles under normal conditions. Therefore, it needs a signal from our brain, from the nervous system, in order to contract. So we're just looking at an end of a neuron. This is called the axon, which we'll start covering on Wednesday. Wait. Yeah, which we'll go over, sorry, on Thursday in the review session when we kind of get to the nervous system. But regardless, here's the end of that neuron, that axon, and it's going to synapse, which is join or come close to joining the muscle fiber. So you see that little lightning bolt that's saying, yeah, that nerve impulse has reached the end of the axon, and that's the message. So now we enlarge it. So here's the axon terminal, which is the end of the neuron, the nerve cell. There's the message coming in, the lightning bolt. And here is our muscle fiber. There's the sarcolemma, which is don't confuse that with the sarcomere. This is the sarcolemma, the plasma membrane. But when the plasma membrane gets close to the synapse here, where neuron meets muscle fiber, it starts to wave in and out. Right? We get this curvy pattern to it. That's called the motor end plate. Right? So the axon terminal of the neuron, nerve cell, is meeting the motor end plate of the nerve fiber, or the muscle fiber, sorry, which is the muscle cell. So the two cells meet right here, but there's a space in between the two, and that's called the synaptic cleft. So there's the space. If we enlarge it, we can see, oh, there's axon terminal. There's the motor end plate, that plasma membrane that curves in and out. And so let's kind of run through these steps here. I wouldn't say you need to memorize these steps, but you should kind of know what happens first, second, third, as we run through the story on how muscle contracts. So this is just kind of the first part of the story. So the action potential, the message that's coming from the brain, arrives at the, synaptic end bulb. That's where that axon terminal kind of gets big at the end. You can even say it might be simpler. The action potential arrives at the axon terminal, which is the end of the axon. Right? It's written right there, axon terminal. So when that happens, well, calcium happens to rush in. You don't see it pictured there, so I'm not too worried about the calcium steps there, these ones. But when that action potential arrives at the axon terminal, all these little vesicles filled with these chemicals gets released. So there's some exocytosis of these chemicals. Now the chemicals are neurotransmitters, and neurotransmitters are little chemical messages that go across a synapse, right? This is a synapse where these two cells are communicating. So in a way to get that message, that action potential, the way to get that message across to the muscle fiber is to send these chemical messages across the gap, across the synaptic cleft. So the neurotransmitters, the chemical messengers that we use in skeletal muscle is called acetylcholine. And it doesn't actually say it there. Does it spell it anywhere here for you? No. I'll write it in just so you can see the actual non-abbreviated name. So acetylcholine, right? And it's just abbreviated often to ACH, just like right here. Right? So the action potential arrives at the axon terminal. That causes the release through exocytosis of acetylcholine, which is a type of neurotransmitter. Let me get my laser. And acetylcholine will then bind to a receptor. You can see these receptors in the motor end plate. There's another one. There's another one. So when acetylcholine binds to the receptors, that opens up a channel. Now you have ions flowing in and out through the channel, which changes the voltage on the motor end plate, but that's fine. So sodium happens to rush in, the motor end plate changes from a negative to a positive, that's fine. And then we get something called an end plate potential. That means all of a sudden, this muscle fiber, right, the sarcolemma or the motor end plate is excited. We learned that muscle fibers are excitable, one of the 4 properties at the beginning of this chapter. Well, now it's excited, and we call that the end plate potential. So the end plate potential is this voltage that's occurring across the motor end plate now. And that's happening because the neurotransmitters bound to these receptors on the motor end plate. So that's an important point, right? Acetylcholine, the neurotransmitters, come from the axon terminal. There are these messengers, but the receptor for them is on the motor end plate. And that binding of acetylcholine to its receptors is what causes the channels to open, the ions to move, right? Ions are charged chemicals. This happens to be sodium. The sodium is a plus charge. I'm not sure why there's a negative there, but that's fine. And we now have an excited motor end plate. So that's called end plate potential. So this is kind of step 1, even though it seems like there's a million steps there. What happened, message came from the brain, and at the end of this slide, well, now the motor end plate is excited, end plate potential. So we got the message across from this cell to this cell, from the neuron to the muscle fiber. So this end plate potential, right, it loses strength as it travels, but the point is we need that end plate potential to reach the sarcolemma. Remember, the motor end plate is the curvy part of the sarcolemma, but on either side here is just regular sarcolemma. So if that end plate potential reaches the sarcolemma, we just say it does, that causes an action potential. An action potential is all or nothing. Once it starts, this action potential, this nerve impulse, the excitability is going to flow through the entire muscle fiber. So just to kind of summarize, end plate potentials loses strength as it travels, but if the end plate potential is strong enough at the sarcolemma, here or here, now we get an action potential, and that's going to go along the entire sarcolemma. An action potential does not lose strength, so once that excitability hits here or hits here, it's like dominoes. We get this voltage across your entire muscle fiber, which we don't see on this picture. It's just a very tiny, right, area on it. All right, so there's a sarcolemma. Let's say it got excited, so that charge is going to that action potential is going to rush through the entire sarcolemma all the way around the muscle fiber. There's a sarcolemma, which is that plasma membrane. But if you look, the sarcolemma, we saw this in lab, has all these little, I call them like little wormholes in them. So when that action potential rushes across the sarcolemma, it also goes down these, what are called transverse tubules or T-tubules. And that allows that impulse, that electrical impulse, to totally surround the myofibrils. So that impulse is action potentials entering the cell on these transverse tubules. There it is, T-tubules, periodic invaginations of the plasma membrane, sarcolemma, into the muscle fiber. So before we finish the story, nothing's contracted yet, right? The muscle fiber is excited. It's got action potential rushing through it, but those myosin and actin have not slid against each other yet. All right, so in order to finish the story, we have to name a couple more players. Sorry, it probably didn't sound good. A couple more players or structures in the muscle fiber. And that's the sarcoplasmic reticulum and the terminal cisterna. So all of these tubes, we labeled them in lab. Those are sarcoplasmic reticulum. They store calcium. That's their job, store calcium. the sarcoplasmic reticulum gets wider along the T-tubules on either side, parallel to the T-tubules. That's just in large parts of the sarcoplasmic reticulum. So there's a lot of calcium stored in the terminal cisternae. So there's a terminal cisternae. There's another terminal cisternae with the T-tubule in between them. So together we have this triad, the T-tubule between two terminal cisterna. So as the action potential rushes down a T-tubule, the calcium stored in there in the sarcoplasmic reticulum or terminal cisterna gets released. Doesn't say that there yet, but that's what's going to happen on the next slide. All right, so this is an information-packed slide right here. There's the beginning of our story, which we already went through. Action potential reaches the axon terminal. Acetylcholine binds to a receptor on the motor end plate. Then we have an end plate potential. And if it gets to the sarcolemma here, now we have an action potential. There's the arrow. And that action potential goes down the T-tubules, right? Then down the next T-tubule and so on. Now here's our sarcoplasmic reticulum. It looks a little different on this picture. But when the action potential travels down the T-tubule, calcium that's stored in there gets released. And what's the calcium? What do these calcium ions do? They bind to troponin. Remember, one of those blocker proteins. Troponin are those little round things. That moves tropomyosin out of the way, the long yellow thing or orange thing. Now, all of a sudden, we can see the binding sites. So now the myosin head can bind to actin. It's called a cross bridge, and produce a power stroke, which is sliding the actin towards the center of the sarcomere. Right? So if we enlarge that, we can see what happens. So the whole point is our muscles are not able to contract because myosin cannot normally bind to actin because of the contact inhibition of troponin and tropomyosin. And that's a good thing. If not, our muscles would be contracted all the time and we wouldn't be able to relax them. So in order to move troponin and tropomyosin out of the way, calcium needs to bind to troponin. Calcium is stored in the sarcoplasmic reticulum until we want to contract our muscles. So when calcium is released, now everything, all that blocking gets removed and we can perform the cross bridge and the power stroke. So that's a big story right there. Go over it a few more times. You should know what happens before something else if you're asked for an order of things. But one more really important point on this slide here. It's something called the latent period, L-A-T-E-N-T. So what happens is the time it takes for the muscle fiber to get excited, we know that happened right here, right? Excitation, meaning neurotransmitters, the acetylcholine, end plate potential, but all of a sudden we have an action potential right here. So it's charged, it's excited, but nothing's contracted yet. So it takes time for all these steps to happen. So the time that it takes from when it's excited to when it actually contracts, the power stroke, that's called EC coupling, right? Excitation, contraction coupling. It's almost instantaneous, right? It happens so quickly, but there is time that it takes for that to happen because the action potential has to go down the T-tubules. Calcium needs to be released from the SR. Calcium has to bind to troponin. That moves tropomyosin out of the way. Now that binding sites are exposed, the myosin head binds to actin, and now we have the power stroke. So the time it takes for that is called EC, or excitation contraction coupling. Of course, just as quickly as calcium is released, it gets pumped right back in, and that allows us to relax our muscles shortly thereafter. When calcium gets pumped back in to the SR, sarcoplasmic reticulum, well, now we can see there's contact inhibition all over again, right? Tropomyosin's in the way, that myosin head can no longer bind. Right, and we've seen that already. Big point here, you can kind of work through this. In my videos, I work through all of this, but what we haven't mentioned that I'll pull from this slide is that this requires energy. The power stroke requires energy, so of course we need ATP. But something that's not so obvious is after the power stroke occurs, so you can see right here that myosin head performed a power stroke, so you can see the arrows, the actin is sliding towards the center of the sarcomere. It's not able to release. It's stuck. It's not until ATP binds to the myosin head that releases it. Right? So ATP drives the process, but ATP also allows the myosin head to be removed from actin. Right? So there's the two roles of ATP. We need ATP for energy and also to release the head from actin. Not too worried about that slide or that slide. A couple more points that I kind of want to bring up here is that what a motor unit is. So a motor unit is one neuron. It's one nerve cell. But you notice how it's synapsing with more than one muscle cell, more than one muscle fiber, showing 5 right here. But the whole thing is called a motor unit. So as long as there's just one neuron, it doesn't matter if it's synapsing with 5 or 5,000 neurons, it's still one motor unit as long as just one neuron sending the signal. So how do we get a stronger contraction? Because this action potential either happens or it doesn't, I can't have a stronger action potential. One way for a stronger contraction of your muscle is to use more than one motor unit. So right here, here's an axon of one neuron. That's one motor unit. Well, here's the green axon of another neuron. So this is a second motor unit. So that's called recruitment. Right? For a stronger contraction, we recruit simply more motor units. That's fine there. Yeah, there's the latent period I was talking about. So the stimulus, the muscle is excited right here, but you notice that it takes a few milliseconds before anything starts contracting. Well, that's the time it takes from when the muscle is excited to when it actually contracts. Excitation, contraction, coupling is our latent period. So then we have contraction, and then we have relaxation. And notice it takes longer for it to relax than it does for a sarcomere to contract. Because of this, our nervous system can do something to make a stronger contraction, is that since it takes so long to relax, when a muscle begins to contract, there it's starting to relax. Why not send another signal in to make it contract again, then again, before it's able to relax totally? So we have muscle contracting, now it's partially relaxing, and then it's stimulated again. So we have the same amount of contraction as here, but there was more, it hasn't relaxed all the way. So we can send action potentials in to our muscle fibers one after another. I'm going to go to the next slide because the picture's better, but it's the same information. Right, same information. So on partial relaxation, another stimulus, like you can see with this arrow, is sent. Right, so each time a stimulus is sent, we get a stronger and stronger contraction because one's just piggybacking on the other. So it's taking advantage of, relaxation takes a lot longer, right? So before a muscle can completely relax, we can build on that contraction, increasing the tension in the muscle. This is called wave summation, and summation means adding on each other. So we get these waves because one contraction's added to the next, right? That's called wave summation. But since it's partially relaxing in between, and by the way, this happens too fast to feel any relaxation, but because it is partially relaxing, we call this, I'm going to go to the slide before, unfused or incomplete tetanus. That has nothing to do with a tetanus shot, by the way. Just means muscle contracting. So unfused or incomplete, that means, we got a little bit of relaxation in between the summation, the wave summation, just like that. But what if we don't wait so long? What if we send another action potential in before it has time to relax, like right there? That's still wave summation, but this is called fused or complete instead of unfused or incomplete. So you see a nice smooth rise in the tension. I'm going to use the picture on the next slide, but it's the same information. So see how close together the muscle fiber is being stimulated by the nervous system. So it doesn't have time to relax. There's a little latent period for AC coupling. It starts to contract, but before it can even begin to relax, another it's stimulated again, right? Then again, right? So you get this complete or fused tetanus. And then there's going to be a point where it can't contract anymore. It gets to maximum contraction. So it doesn't matter how many more stimuli you bring in. Once you're at max contraction, you're at max contraction. And then once there's no more stimuli, it goes and through its relaxation period. All right, so muscle tone, just a quick thing. I wouldn't worry about the second half here. It's just that our muscles are always partially contracted, even when we believe them to be at rest. And this makes them ready. There's always some overlap between actin and myosin. So if we do need to contract a specific muscle, that overlap of actin and myosin, so those myosin heads can quickly grab onto actin and produce an adequate contraction. So muscles remain in partial state of contraction. You see that when if someone has a stroke and their face droops, right? Now they've lost that partial contraction, right? Versus the tone of a normal muscle overlap, or I should say, myofilament overlap between actin and myosin. Don't need to go over that. Or cardiac muscle. So the last point that we would need to go over here, I think would be helpful, is we just went through a massive story on how muscles contract, right? From the nerve impulse all the way until the power stroke, the EC coupling, all of that stuff. That's skeletal muscle. We also have smooth muscle, which is our organ muscle, which is different. There's some similarities, but there's some differences as well. Well, you could, of course, read through that, but there's no neuromuscular junctions like skeletal muscle has and smooth muscle. Instead, we have something called diffuse junctions, and we have something called dense bodies first. So instead of Z discs, there's dense bodies and smooth muscles, and that's all these little nodes that you're looking at here. So in addition to actin and myosin, which are also in smooth muscle, we have another myofilament, and they're called intermediate filaments, and that's those strings that you see here between dense bodies. So when actin and myosin slide against each other, it pulls on the intermediate filaments, which pulls these dense bodies sort of in a corkscrew manner, wringing out, right, the smooth muscle that's in our organs. So dense bodies instead of Z discs, and in addition to actin and myosin, smooth muscle has one more filament, myofilament, called intermediate filaments. So instead of a neuromuscular junction, we have something called diffuse junctions because it's just very general. So instead of the axon terminals, we have these varicosities. And varicosities are these little swellings on the axon. So when an action potential travels down the axon, the varicosities release their neurotransmitters, kind of spread it along the entire smooth muscle, just kind of sprinkling all the neurotransmitters on. So very diffuse. The neuromuscular junction in skeletal muscle is very specific. It's a very specific connection, but here it's just a lot more general, just kind of diffusely, for lack of a better word, right? These diffuse junction across a layer of smooth muscle, right? And another important point here, I'm not going to go over a single unit or a multi-unit. That's an easy read, but one more important thing I do want to go over with you guys here before we go is calcium and its role compared to skeletal muscle. So in skeletal muscle, we store calcium in the sarcoplasmic reticulum. And then when it's released from the sarcoplasmic reticulum, it binds to troponin, and that removes the contact inhibition. So we covered that with skeletal muscle. But in smooth muscle, yeah, some calcium is in the sarcoplasmic reticulum. There's a little bit in there, but most of it is outside of the cell in these little caves called calveolae, little caves. So if I look at smooth muscle, they exaggerated the size of them here, but you can see these little calveolae, little caves, and calcium kind of resides in those calveolae. So when the diffuse junction, which you don't see here, comes in and spreads the neurotransmitter over the smooth muscle, well, now calcium enters the cell. Some gets released from the SR, like skeletal muscle, but most calcium comes from the calveolae from the outside. That calcium in skeletal muscle binds to troponin, but in smooth muscle, we got the calcium coming in from the outside. It binds to calmodulin. So there's a difference. So instead of binding to troponin, it binds to calmodulin, and that will go through a series of reactions, which you can save yourself. But the end story is, by the end of these reactions here, actin and myosin slide against each other. So the reason why I paused here on this slide and kind of went a little slower is because this is a common mistake. I don't know, maybe because both words start with the letter C. Calveolae, calmodulin. Difference between smooth and skeletal muscle. Well, in smooth muscle, calcium is stored outside in the calveolae, and when calcium enters the cell, it binds to calmodulin and not troponin, which will eventually cause actin and myosin to slide. So there it was, the main parts of chapter 10.